Welcome everyone to the Students Presentation Week for the academic year 2025-26. This special event celebrates our collective efforts and commitment to learning at the PHARMACOGNOSTIC Institute of Pharmacy. I am Mr. Nihal Uprikar from B Pharmacy 5th semester, roll number 30525, and I am excited to take you through an in-depth look at the pharmacognostic study of SENNA, a remarkable medicine plant that plays a vital role in pharmacy and traditional healing. Before we begin, let me give you an overview of what we will be discussing today. Our presentation will journey through the various facets of SENNA. We will start by uncovering its synonyms, the different names by its recognized. We will then investigate its biological and geographical sources, dwell into the details of its cultivation and collection, and explore its microscopic or visible as well as macroscopic characteristics. Further, we will discuss its rich chemical compositions, examine how these compounds are extracted and isolated, and learn about the specialized chemical test known as bond trigger test that helps identify these compounds. We will touch on the method used to assay or evaluate SENNA, recognize common adult trends and substitutes, and finally understand its primary uses in medicine and some of its marketed pharmaceutical products. By the end of this presentation, you will gain a comprehensive appreciation of SENNA's significance. Let's start by introducing the primary compound of interest in SENNA, anthracene glycosides. These are naturally occurring organic compounds found predominantly in dicot plants, with SENNA being one of the most prominent examples. Other plants such as aloe, rhubarb, and cascara also contain this active substance. The backbone of these glycosides is anthraquinone, an aromatic organic compound derived from anthracene. This structure acts as a foundation for many plants' pigments and impart characteristic physiological effects. To identify these glycosides, a specialized chemical test called bond trigger test is performed, which produces a distinctive color reaction, an essential tool in pharmacognosy. Now, turning our attention to the SENNA itself, let's address the various synonyms by which it is known. In different regions and by different traditions, SENNA may be referred to as Alexandrian SENNA, Tineveli SENNA, and Folia SENNA. This name reflects its diverse heritage and broad medicinal usage across cultures. From a biological perspective, SENNA leaves mainly consist of the dried leaflets of two species, Cassia acatifolia, commonly known as Alexandrian SENNA, and Cassia augustifolia, known as Tineveli SENNA. Both belong to the Leguminaceae family, which is celebrated for its range of medicinal plants. Each type has subtle difference in morphology and chemical compositions, influencing their commercial and therapeutical application. SENNA is geographically widespread, growing in both arid and semi-arid climates. Its main source includes the African continents, specifically South Africa, Egypt, and Sudan. In India, SENNA is predominantly cultivated in Tamil Nadu, Punjab, and parts of the Sindh region. The plant's lifespan ranges from 2 to 3 years. It prefers clayed, semi-irrigated soil, thriving where water retentions and drainage are balanced. In the field, leaves are typically harvested 2 to 3 months after planting, time to collect the most potent raw materials. Alexandrian SENNA is often collected from wild populations, while Tineveli SENNA benefits from controlled cultivations, helping maintain purity and quality. Examining SENNA at the macroscopic levels reveals several unique features. The leaves are a yellowish-green color, signaling their freshness and good quality, with characteristically slight odor. Upon tasting, one finds a bitter and mucilaginous texture, a hallmark for the authentication of SENNA. The shape of SENNA leaves is lanceolate, meaning they are long and narrow with an acute pointed apex, frequently displaying a small spine at the tip. Each leaflet is asymmetrical, and fine transversal lines can be observed, along with the presence of trichomes or tiny hairs on both surfaces. These features are important for visual identifications, particularly to distinguish authentic SENNA from possible adulterants. A closer microscopic inspection of SENNA leaves uncovers further diagnostic details. The leaf is isobilateral, having pellicate tissue on both the upper and lower surfaces. The epidermis is composed of unicellular, conical, thick-walled trichomes with a warty appearance, a trait that assists in identification under the microscope. These trichomes are curved and are present on both sides of the leaves. Of particular note is the type of stomata, parasitic, which is found on epidermal surfaces. Deeper within, one can find sclerenchymatous fiber for support and bundles of xylem and phloem, as well as calcium oxalate crystals and spongy parenchyma. Each structural detail contributes to the leaf's mechanical strength and defense while serving as features for authentication. The chemical makeup of senna is what makes it so valuable in medicine. The plant contains less than 2.5% of two principal anthraquinone glycosides known as senoside A and senoside B. These are stereoisomer mirror images of each other, where senoside A is dextrorotatory and senoside B is meso. In addition to these, the plants includes other important constituents such as rein and aloimodine, both known for their medical potential. Volatile substance, mucilage, resins, and water-soluble polysaccharides are also present, contributing to senna's wide range of therapeutic effects and facilitating its clinical formulation and extraction. To harness these potent compounds, senna leaves are subjected to various extraction and isolation techniques. The powdered drug is treated with either 90% methanol or 80% acetone for several hours, followed by cold water extraction to maximize the yield. Remarkably, these extracts can contain up to 17 to 18% cyanoids, indicative of high quality plant materials. Alternative extractions method use non-ionic surfactants combined with polyethylene glycol in 70% ethanol to efficiently extract senosides and related derivatives. For isolation, further treatment with citric acid in methanol and subsequent extraction using a methanol toluene mixture, ammonia and precipitation with calcium chloride is performed. Non-polar synthetic resins with porous structures are also proposed for industrial scale isolation, paving the way for highly pure senna-based pharmaceuticals. A fundamental step in verifying the presence of anthraquinone glycosides in senna is the bond trigger states. Let's walk through the process. The drug is first boiled with dilute sulfuric acid and then filtered. The resulting filtrate is mixed with an organic solvent, typically benzene, ether or chloroform, and shaken well. Upon separation, ammonia is slowly added to the organic layer. If anthraquinones are present, the ammoniacal layer turns a distinct pink to red, confirming both the identity and purity of the senna extract. This straightforward colorimetric state is invaluable for the quality control in medicinal plants research. The quality of senna must not be verified but also quantified, ensuring accurate medicinal doses. Assays are performed using both biological and chemical methods. On the chemical front, thin layer chromatography, spectrophotometry, and spectrocolorimetry are commonly employed to determine senoside content. Biologically, the efficacy can be measured by counting the number of weight phases produced in test animals, such as mice, after administration, indicating the drug's purgative power. It's important to be vigilant about the adulterants that can be compromise efficacy and safety. Common adulterants and substitutes include dog senna, pal senna, and variants like Bombay, Mecca, and Arabian senna. These may differ significantly in chemical compositions, so carefully examine is vital prior to use in therapy. Senna's uses are extensive and well recognized in modern and traditional medicines. Its primary role is a purgative and cathartic, efficiently relieving constipation through its action on the intestines. Additionally, senna is used to ease biliousness, aid digestion, and is sometimes considered beneficial in managing symptoms of irritable IBS. Its dual action as both a gentle and effective remedy is why senna is found in a host of marketed products. Many international pharmaceutical brands like Syned, Persenid, and Xlax contain senna as their core active ingredient. These products, available in various formulations, attest to senna's enduring value and regular use as a trusted household remedy. The widespread adoption of senna-based formulations by reputable manufacturers such as Cipla, GSK, and Novartis further reinforce its global acceptance. These brands ensure quality and adherence to safety standards, giving consumer peace of mind and effective relief through consistent and standardized products. As we approach the conclusion of this presentation, I would like to express my heartfelt gratitude to my esteemed guide, Dr. Nilesh Banarse, whose continuous support, motivation, and guidance has been invaluable in preparing this work. My sincere thanks also extend to the respected principal, the director, all faculty members, and the dedicated non-teaching staff for their encouragement and assistance. A special note of appreciation goes to my friends and classmates whose cooperation and support have meant so much during this academic journey. Thank you all for your attention throughout the detailed exploration of the pharmacognostic study of SENNA. I hope this presentation has provided you with meaningful insights